Hey everyone, welcome back. In this video, we're going to upload real sequence data to our cloud server and run the NF-core RNA-seq pipeline. Let's get started. This dataset comes from a yeast RNA-seq experiment comparing normal cells to cells where the transcription factor RAP1 has been degraded. And for this demonstration, we'll use a subset of the samples from both the control and experimental groups to show how to upload and run real RNA-seq data through the NF-core RNA-seq pipeline in the cloud. And if you'd like to learn more about this experiment, I'll include a link to the bio project in the video description. I've already downloaded the raw sequence data to my personal hard drive, about 25 gigabytes of raw data, along with the Saccharomyces cerevisiae reference genome, yeast, and gene annotation files, which are in FASTA and GFF format. I've also included a couple of additional files that we'll need to run the pipeline, a sample sheet and a resource configuration file. And I'll explain these in a little bit. To transfer the data to our cloud server, we're going to use a command line tool called rsync. And what rsync allows us to do is efficiently transfer files between computers, which makes it ideal for moving large sequencing datasets. To initiate the transfer, we'll need the username, IP address, and password of our remote server, and we can find this information in our Vultr server dashboard. So here's the command I'm using, and yours might look slightly different depending on your server. And what this does is tells rsync to transfer everything in the current working directory of my hard drive to the data directory on our remote server, which you'll need to create, but you can call it anything. And for large data sets like RNA-seq data, this transfer can take some time. Once the files are on the server, we can move into the data directory. Inside, we have the FASTQ files containing the raw RNA-seq reads, a GFF file containing the gene annotations, a FASTA file containing the reference genome, a resource configuration file, which defines how many computational resources the pipeline is allowed to use. For me, I'm running a server with 16 CPUs and about 32 gigabytes of RAM, so I don't want the pipeline to exceed these limits. And if you noticed, I actually put 30 gigabytes here for memory instead of 32 gigabytes, simply because the server is already using some memory by default, so I need to account for that. And finally, I have a sample sheet, which describes the samples and experimental design. And the sample sheet is something that you need to create yourself based on your dataset and the pipeline you're using. Different NF-core pipelines will have slightly different formats for the sample sheet, and this is something you can learn more about in the documentation. But the basic format requires a column with sample identifiers, columns specifying the location of the data on your server, and a column specifying the strandedness of your libraries. And before running the pipeline, I'm going to start a screen session, which allows the pipeline to keep running even if we disconnect from the server. And I'll call this screen session Run 1, but you can name yours whatever you like. We can use the ls command to show the name of the session, when it was created, and whether we're attached to it or not. Now we can run the pipeline. And once we run this command, Nextflow will start executing the pipeline steps automatically. I can then detach from the screen session by typing in Command A and D, and then use the ls command again to make sure I detach from the session properly. And if we want to attach to the screen session again, we can use the -r command with the name of the session. Now this run will probably take a few hours to complete depending on the server resources and the size of the dataset. In the next video, we'll look at the results of our analysis and show how to save and download the output files from our cloud server back to our personal hard drive. I'll see you there.